Yeah.
The question that we're trying to answer is what the effect of adding a base to a solution with phenolphthalein in it, and what the surface area would need to be for that change to be observable.
Okay.
So our group had the shark-shaped cutout for our phenolphthalein gel, and when we put it in the solution, we used 2 sharks to kind of get more of a baseline, pick up more surface area. And we put the acid in, and it took a while to kind of get going, but it got clear on the edges really quick because the shark had a high surface area, lots of curves. But our gel was really thick compared to other groups, so it took a while for the very middle to turn all the way clear. But at the very end, like right before class ended, it had gotten all the way clear with like one pink dot in the very middle where it was thickest.
Okay, so our gel was shaped like a penguin, and so it had a large amount of surface area because of all the curves. As a result, our gel turned from pink to white quicker. Although at the end we did have a large amount of pink still in the center, but all the edges were completely white.
Our hypothesis was that the potato with the most mass loss after the period of time would be the one that was in the most concentrated solution.
The materials we used for our potato lab project started with 6 beakers with 6 different solutions that were placed into them, 6 potato slices, 1 potato board to cut our slices. We had a scalpel to cut them down to size, tape to tape and label our beakers for each solution, a ruler to make sure each potato slice was the same size, and then a scale to measure or to weigh the solutions and the potatoes. The procedure for this lab started with labeling the 5 beakers, A through E, and then we also had a beaker labeled for water. We then used the potato board to cut equal sizes of potato. We then weighed each piece of potato and then cut them down to the same length. So our weight was exactly the same, and our measurement, we did our best to measure them exactly 5 centimeters long. We put 1 piece of potato into each solution, so we had 6 pieces, and then we poured 50 milliliters of solution into all of the beakers, and then we let the beakers sit overnight. We then removed the potato slices from each solution and measured them and weighed them, and then compared our data.
So the weight of the original potato pieces that were put into all 6 solutions were 0.160 grams, and they were all 5 centimeters long and put into 50 milliliters of solution, just enough to cover the piece of potato entirely. After the 24-hour waiting period, the weights for solution A, the potato in solution A, was 0.130 grams, and the length was 4.5 centimeters. After our calculations, we figured out that it was the 0.6 molar concentration with a water potential of 29.5. Solution B ended up being 0.165 grams and 5 centimeters long, with a 0.2 molar concentration solution, and the water potential was 9.8. In solution C, the mass ended up being 0.115 grams with 5 centimeters length, and 0.8 was the concentration of the solution, with 39.3 as the water potential. For solution D, the mass also went to 0.115 grams and 4.5 centimeters, which is the largest one. So we decided it as being the 1 molar concentration, and the water potential ended up being 49.1. And for the last solution, it was 0.155 grams and 5 centimeters long with a 0.4 molar concentration and a 19.6 water potential. And for our constant, it ended up being 0.175 grams, which was a 0.015 gram increase from the original weight, and was 5 centimeters long after the reaction.
When introduced in an acid, phenolphthalein turns from pink to clear, while in contact with a base it turns bright pink. Solution B had the lowest water potential, which meant that the water and the solute in both in the plant and in the solution were both equal, and not diffusing or causing osmosis to happen. If placed in a dry area for several days, it's possible that the experiment wouldn't work due to rotting or excessive dehydration of the plant. This experiment could only be done with plant and fungal cells because they can produce their own food through photosynthesis and therefore can complete cellular respiration, while animal cells are only capable of surviving when attached to the original organism, which will provide them with sustenance and energy to basically fulfill cellular respiration. Our group matched the solutions perfectly and accurately, although possible errors could have arisen from our cutting of the potato pieces due to small pieces being needed to be shaved off to keep the mass constant between all pieces of potato. And one other flaw is how it affected the plant, because it's possible that the cellular functions that were taking place in each solution were possibly different because of the different concentrations of phenolphthalein, which could have a negative or a positive effect on the ability for the plant cells to either produce food or react with cellular respiration and with their environment.